Hi everyone, and welcome to Chapter 12. This is the first of two lectures on Mendel's experiments and heredity. So our objectives in this first lecture are to explain Mendel's principle of segregation, describe the outcome of a monohybrid cross, be able to work a monohybrid Punnett square, explain Mendel's principle of independent assortment, and describe the outcome of a dihybrid cross. So heredity defined is a passing on of traits from parents to offspring. You're familiar with the term inheritance, where offspring inherit traits from their parents. So familial characteristics were recognized many centuries before today. And in this little illustration over here, you can clearly see some of the phenotypic traits that have been inherited by the two children from their father. Namely, here the red hair, the shape of the face, the shape of the eyes, and my little note says here, any similarities between the humans and the cat are completely coincidental because the cat also has red hair. But the scientific basis for those similarities were non-existent. People just didn't understand why those similarities existed between parents and their children. Now, what was recognized is that heredity occurs within species and the traits are transmitted from parents to offspring. And it was thought the characteristics were blended from generation to generation. But that, of course, is not always true. So the paradox is if blending occurred, then all individuals of a species would eventually look the same, right? Because over time, if the traits, if the characteristics, if the phenotypic characteristics blended, then eventually you get similar members, similar individuals of a species mating, and of course their offspring are going to be very similar, kind of like that happens in bacteria. But of course that doesn't happen in eukaryotic species. So that was the big question centuries ago. So we have another term here, hybridization. And a hybrid, think of the hybrid vehicles now, right? Sometimes it can run on gasoline, sometimes it can run on electricity. It's a hybrid, it's a mixture of the two. So hybridization occurs when you cross-fertilize different strains of a species, and we call these the parent strains. We'll talk about Mendel here in a little bit, but he used the garden pea, Pisum sativum, and it was a model organism then, and it still is, but it was a model organism very early on because its traits are binary. They're cut and dry, right? They're black or white. They're round seed or wrinkled seed. They're purple flower or white flower. There's no in between, no mixing. And of course, pea plants have a short generation time, so it was easy to grow multiple generations in a single season. So T.A. Knight, so Joseph Kohlreuter, I know I'm tearing his name up, in the 1700s, late 1700s, cross strains of tobacco plants, and he produced fertile offspring that were different from both parent plants. See, this work on heredity, even though they didn't have the terminology, was going on a long time ago. T.A. Knight in 1823 performed crosses using garden peas. Mendel would later pick up on that work. So Knight crossed true breeding green seed plants with true breeding yellow seed plants. And these plants represented the parental generation, or just for short, we call it the P generation. Now the progeny, or the offspring, of that of that crossing, of that breeding, if you will, the green seed plants with the yellow seed plants yield the progeny or the offspring. And the progeny in this little simple cross here yielded all yellow seeds. Now, of course, these were hybrids because they're a mixture of green seed plants and yellow seed plants. But the first generation, we call it the F1 generation. That stands for filial generation, first filial generation. Yielded all yellow seeds. All the plants of the first generation had yellow seeds. So then the question was, what happened to the green seeds? Because one of the parent plants had green seeds. But when the F1 plants, take two plants from the first generation with all yellow seeds and you cross them, a mixture of yellow seed and green seed plants resulted. And so these plants represent the F2 or second filial generation. And we'll see how that works out here in a little bit. So in this first little video that I would like you to watch is just a simple introduction to genetics and heredity. None of these videos, as you know, are that long, but you'll pick up some terminology in that little video. So we have Gregor Mendel. He's considered the father of modern genetics. And he would continue Knight's hybridization experiments with pea plants at his monastery, the monastery where he lived. He was a monk. And so he took the pea plants. He used the pea plants because it was an ideal organism for several reasons. Hybrids could be produced, green seeds crossed with yellow seeds, purple flowers crossed with white flowers. There are many varieties of peas. They're small and easy to grow, don't take up a lot of space, and of course, living in a monastery, there's not a whole lot of ground to work with. They can self-fertilize or they can be crossed. And of course, peas are good in a salad, so he could feed the brethren with the results of his experiments. And so he produced true breeding strains for each trait that he wanted to study, and we'll look at the 7 traits that he studied in a little bit. The parent or purebred or the pea generation, as I've mentioned. Cross-fertilized true breeding strains having alternate forms of a trait, produces the F1 or the first filial generation. Alternate forms of a trait. So seed color, right? We have seed color and we have two alternate forms, and we call them alleles. One is green seed and one is yellow seed, or purple flowers with white flowers, or tall plants with short plants. And so crossing the purebred parents yields the hybrid F1 generation. And then if you allow the hybrid offspring to self-fertilize for several generations and count the number of offspring showing the form of each trait, then you get very definitive ratios. And we'll look at that here in a minute. So here is Mendel's methods, as it were. And so, this is just a pea plant with its purple flower. So here he's taking one part of the purple, the pollen here of the purple flowered plant, and he's fertilizing the plant that produces white flowers. And in the first filial generation, all the progeny end up with purple flowers. And so his question was, well, what happened to the white flowers? Because I crossed purple with white. Here there's no blending, there's no light purple. And there is no demonstration of white flowers. So that brings us to the monohybrid cross. Now, mono means one, of course, and we're talking about, in this instance, one trait, flower color, or seed color, or the height of the plant, or wrinkled seeds versus smooth seeds. So one trait, that's the mono part, and it's hybrid. So we're crossing two different alleles, or two different variations of the same trait. So we have, if the trait is seed color, we cross green seed plants with yellow seed plants. See, that's the hybrid part. The F1 generation, of course, are the hybrids. So monohybrid cross is an experimental cross that examines only two variations of a single trait, like flower color, seed color, plant height. And Mendel produced true breeding pea strains for seven different traits. Okay, here they are: flower color, seed texture, that's smooth or wrinkled, pod color, that's green or yellow, pod shape, flower position, is it on the is it a terminal flower? In other words, is it at the end of a branch, or does it fall somewhere in the middle? That's flower position. And plant height, tall versus short. And each of these traits has two variants, and those variants we call alleles. And here they are. Here's his top 7 traits that he looked at. Pod color, pod shape, or the pod shape, it's either a smooth, they call it inflated, but it's smooth, and here it's bumpy or constricted. It's constricted around where the actual peas in the pod are. So you can see the texture there. I'm not going to ask you, which of the following traits did Mendel study, and give you a series of traits there, but it's the idea that he studied 7, he made true breeding parents out of them, and he crossed them, and that's where his experimentation and his results came from. And so you can see that when he did this, so the number here, the total number down here, are the total number of offspring that he ended up with. And then what he did, right, this has purple flowers crossed with white flowers. He actually physically counted them. And out of the 3,000 plus, he ended up with 700, just over 700 purple hybrids yielding purple flowers, and just over 200 hybrids yielding white flowers. And the same played true with all of these traits. There's 600 yellow, 2,000 yellow, 6,000 yellow, 2,000 green. Down here we have almost 3,000 plants, and you can see the split there. And in all of these, what I want you to notice here with these numbers is that you roughly have a 3-to-1 ratio. Okay, so this is 3.15 to 1. 3.0 to 1. It's hard for me to see because these are very small on my monitor, but the point here is that you're getting roughly the ratio here, and the F1 generation is roughly, or the F2 generation rather, is roughly 3-to-1. And what that told Mendel, and what you need to learn from this, is that when you do a cross, a monohybrid cross like this, you're going to always get roughly the same 3-to-1 ratio. And we'll talk about what that means here in a second. So the first filial generation is the F1 generation, or F1, the offspring produced by crossing 2 true breeding parental strains. Okay, true breeding, that means purple flowers with white flowers, right? That means if you have two alleles and we use P for flower color, then true breeding is going to be, you got two alleles, true breeding is going to be PP, or rather capital P, capital P. All right, it's going to be the same allele, right? That's true breeding. Okay, for every trait Mendel studied, all of the F1 plants resembled only one parent. And so he referred to the trait here as dominant. In other words, it's the expressed phenotype. Phenotype, of course, is the physical appearance, right? You have red hair, that's your phenotype. You have blue eyes, that's your phenotype. And so the trait that was expressed in the F1 generation was the dominant trait, because somewhere in there, he crossed purple flowers with white flowers, somewhere in there has to be white. There has to be a gene for white. There has to be an allele for white. It's just hidden here. And so the one that gets expressed is he called dominant. And the alternative trait that was, if you will, hidden was he called recessive. So the dominant always gets expressed, and the recessive always yields to the dominant. And so in the F2 generation, now remember F2, second filial generation, is taking two F1 plants and crossing them, or letting the F1 plants self-fertilize. Second filial generation, the offspring are the offspring resulting from self-fertilization of F1 plants, first generation plants. And although hidden in the F1 generation, the recessive trait reappears in the F2s. And then after counting all of the proportions of traits, he always found, and you will always found if you were to do these experiments, roughly a 3-to-1 ratio. That is 3 dominant traits expressed, 1 to every 1 recessive trait expressed. Now, in this video, it talks about, this is from Professor Dave, I think. It talks about Mendelian genetics and Punnett squares, and we're going to go to Punnett squares in a little bit. So it'd be worth your while to watch this video. So back to Mendel. In the F2 generation, 3/4, right, it's 3-to-1, so 3 plus 1 is 4. So 3/4 of the plants displayed the dominant form. Now remember, in the F1, 100% displayed the dominant phenotype. In the F2, 3/4 of them displayed the dominant phenotype. 1/4 displayed the recessive phenotype. And so dominant to recessive ratio was 3-to-1 roughly. That's something you want to remember as we talk about Punnett squares. So Mendel discovered the ratio is actually one true breeding dominant plant that's homozygous, right? True breeding, that's homozygous. Homo, the same, right? Two forms of the same gene. We call them alleles. And we represent those with capital letters. You can use whatever letter you want here. It's conventional to use, for instance, if we're talking about flower color and the dominant flower color is purple, to use purple for flower color. So the dominant purple would be capital P, and the recessive white would be lowercase p. You want to use the same letter for both forms. If it's dominant, capitalize it. If it's recessive, you lowercase it. So true breeding dominant would be big A, and not true breeding dominant plants. Okay, true breeding, two big A's. Not true breeding, we call those heterozygous. See, hetero means different. Homo means the same. So the genes are the same, the alleles are the same, big A. Hetero means different, so the alleles are different And so we represent the dominant one with a capital A, or big A we say, and the recessive one with a lowercase a, or little a. So hetero means different. That means the alleles are different. So big A, little a. Homo means the same. That means the alleles are the same, big A, big A. And so here is Mendel's monohybrid, one trait, cross for plant height. We have tall plants and short plants. So tall is dominant, so we're going to use capital T for tall, lowercase t for short. You could use big X, little x, whatever you want. You can use whatever the letter that you want to pick, but usually by convention, it's whatever the first letter is of the dominant trait. Now remember, okay, so the cross of two heterozygous parents. Okay, parent here is maybe not the best word to use because I don't want you to confuse that with the parental generation, the P generation. Because remember, in this situation, all the parents are true breeding. Big T cross with little t, and then you cross them, and then you get the heterozygotes. We'll see how that works here. Heterozygous or heterozygotes means that you have two different forms of the gene. So we have a tall gene and a short gene crossed with a tall gene and a short gene. So this is the F1 generation, right? So you cross two plants from the F1 generation and you get the F2 generation. And in order to do this, this is called a Punnett square. It's just a little simple box with divided into 4 because we're only looking at one trait. And you're going to put one parent, you're going to put the genotype of one parent up here and the genotype of the other parent down here. And when we get to animals, it doesn't matter if this is the male and that's the female, it doesn't matter. And so you have big T, little t, big T, little t, big T, little t crossed with big T, little t. And it's just a simple matter of cross multiplying, if you will, right? So big T times big T equals big T, big T. Big T times little t equals big T, little t. Little t times big T equals little t, big T. Now here it doesn't matter the order, okay? So I just flip them around so that it looks normal. We usually have the dominance on the left and we read to the right. But you could easily have put little t, big T. It means the same thing, right? 3 times 2 is 6. That's the same as 2 times 3. You're still going to get 6, so it doesn't matter. And here we have little t times little t. That's little t, right? And so let's look at these, right? This is big T. So it's the same dominant allele. So this is homozygous dominant. Likewise, this is little t, two alleles of two short alleles, right? So they're the same. So this is homozygous and they're little, so this is recessive. And then we have a big T and a little t, so this is heterozygous, but phenotypically it's going to be tall because the dominant always dominates over the recessive. And likewise down here we have big T, little t, so that's heterozygous, phenotypically tall, right? So we would say that's heterozygous tall. That means you have an allele for tall, an allele for short, but since the allele for tall is dominant, that's what's going to project in the phenotype. So genotypically, so you want to be able to do a Punnett square and then tell me back what is the genotype ratio and what is the phenotype ratio. It's not always one and the same. So when we think about genotype, we have to look at the alleles. So here we have A big T, we have one of those out of 4. We have one of those, or 25% big T. Here we have 25% little t. And here we have, these are the same, so this would be 50% big T, little t, or 50% heterozygous. Or if we're just talking about the 4, here we have a 1, 2, 1 ratio. 1 to 1 ratio. And you can read it there for you. 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. Now, what's going to happen phenotypically, though? Well, all of these plants are going to be tall, right? Dominant T. These plants are going to be tall. Dominant T dominates recessive T. All of these plants are going to be tall. Dominant T dominates recessive T. And all of these plants are going to be short because we have 2 recessive alleles. We're talking about recessive alleles only when you have 2 copies of the recessive gene can the plant express that recessive phenotype, because dominant always dominates over recessive. Here there's no dominant, they're both recessive, so then you get a recessive. Now, phenotypically then we get tall, tall, tall. Even though these are heterozygotes, they're going to be tall because we have a big T there. So we have 3 tall and 1 short. 3 tall, 1 short. That's a 3 to 1 phenotype ratio, but it's a 1 to 1 genotype ratio. Okay, that's how that works. So the Punnett square, which we just looked at, is a simple way to visualize a genetic cross. Mendel crossed purple flowered plant with a white flowered plant, and these were true breeding parental plants. P is the dominant allele for purple flowers. So we're talking about flower color is the phenotype. Flower color is what we're looking at here. And little p or lowercase p is recessive allele for white flowers. So a true breeding white flower has to have 2 copies of the recessive allele because white is recessive. And the only way a recessive can be expressed is if there are 2 copies. So little p, that's homozygous recessive. Homo because they're the same. True breeding purple flower plant is big P, right? So that's homozygous dominant, homozygous because they're the same. And then big P, little p is the heterozygote. But phenotypically it's going to be purple because the big P is dominant over little p. So here's another look at the same thing, okay? True breeding purple flowers, that's big P, crossed with true breeding white flowers, that's little p, and you end up with, in the first filial generation, the F1 generation, all purple flowers, but you know that they're heterozygotes because you've crossed purple and white. So somewhere in here there has to be a white gene. So that means that these are all the plants, even though they have purple flowers, are heterozygous. Heterozygotes, big P, little p. We're talking about using P for color here. All right, so then in the F2 generation, you take 2 F1 plants, which are heterozygous, and you cross them. Self-cross, self-fertilize, or fertilize them yourself, or however you want to do it. But what you end up with is, phenotypically, 3 purple flowers, or plants bearing purple flowers. You have 3 of them out of 4. And 1 plant bearing white flowers. And what you see here is that in order to be white, it has to be homozygous recessive, little p. And at least one of these three has to be homozygous dominant, big P, big P. And the other two are going to be heterozygous, big T, big P, and little p. Right? And so phenotypically, you get a 3 purple to 1 white. So you get a 3-to-1 ratio. But genotypically, you get 1 big P, 1 little p, and you get 2 heterozygotes. That's what that's representing here, is represented here. Okay, so it's a 3-to-1 phenotypic ratio, and it's a 1-to-1 genotypic ratio or genotype ratio. And this is just another way of looking at it. The F2 generation is heterozygous, big P, little b, big B, little b, big B, little b. And you see, you can, this is for flower color also, you can use whatever letter that you want. By convention, again, we usually use the first letter of the dominant allele for whatever. We're looking at flower color. Conventionally, we would use big P, little p for flower color, big T, little t for plant height, tall versus short, like that, okay? Genotype ratio in a monohybrid cross is always going to be 1-to-1. That's 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. And the phenotypic ratio is always going to be 3-to-1. Remember those numbers. That plays true no matter what trait you're looking at. So here we go. So the parents transmit discrete factors. That's what Mendel called them, discrete factors. We now know they are genes, and each individual receives 1 copy of a gene from each parent. Now, not all copies of a gene are identical, and so we have some terminology here that you've heard already. Allele is an alternative form of a gene. Right, so here we have 2 genes on this plant, right? And if we're just looking at, this is all mixed together, but if you're just focusing on the purple here, right, these genes are the same, so that would be a homozygote. And then here we see the yellow and the green. They're different. Same gene, same location on the chromosome, same gene for whatever the trait is, but they're different forms. Okay, so this could be for green skin versus purple skin if you're talking about aliens. All right, so that would be, that would be heterozygous. Different forms, different alleles for the same gene. So homozygous is 2 of the same allele. That's what you see up here. And heterozygous are 2 different alleles, but for the same trait. So alleles remain discrete. There's no blending in terms of Mendelian genetics. There's no blending here. Alleles remain discrete. So it's either purple or yellow, you don't, or white. You don't get a light purple, right? It's either blue or yellow. You don't get a green in there. Blue and yellow make green, but you don't get that in Mendelian genetics. So the presence of an allele does not guarantee expression, as you now know. If the allele is recessive, it does not get expressed unless there's 2 copies. So a dominant allele gets expressed, a recessive allele remains hidden by the dominant allele. And again, the only way that you get the recessive allele being expressed is if there are 2 copies of the recessive gene. So the genotype is a total set of alleles in an individual carries, and the phenotype is a physical expression of the genotype. All right. So if you have a gene for blue eyes, right? The gene for blue eyes, the blue eyes is the expression of the gene for blue eyes. It's what you see. Phenotype is what you see physically. Now there are two alleles for the gene segregate, you know this already, or separate during gamete formation, are joined at random, one from each parent during fertilization. So we have segregation and random mixing. That's where you get all of the diversity, all of the that we saw in that first picture that I showed you in the last chapter, but also in this one at the very start, with all the children looking up, or all the corn seed, different color corn seeds on the same cob. That's where you get all that variation, because the genes segregate or separate during formation, and then they get rejoined at random during fertilization. The physical basis for allele segregation is the behavior of chromosomes during meiosis, and as you know now, that's anaphase 1. Now, interestingly, Mendel had no knowledge of chromosomes or meiosis, right? That had not been discovered. Nobody had described that during this time. But he realized that something was going on, that these discrete factors, flower color, plant height, pea color, pod shape, these discrete factors, he understood what was going on there, even though he didn't know anything about genetics. Okay, you have any questions? Let me know. And that's the end of the first lecture. So next time we will press on with chapter 12.